Hi everyone, we are group 14 and we'll be going over gliding motility. So why do bacteria need to be able to move? Well, they need to be able to move so that they can go towards favorable environmental conditions that will help them colonize an area, and they need to be able to move away from unfavorable environmental conditions that could be detrimental to their health. There are 4 different types of motility: swimming, twitching, swarming, and gliding. Today we'll be focusing on gliding. Gliding motility is defined as smooth translocation of cells over a surface by an active process that requires the expenditure of energy. Gliding does not use flagella. Instead, it uses pili, which are used in bacterial conjugation. These are some of the different groups of bacteria that exhibit gliding motility. If you remember from class, we've talked about cyanobacteria a lot. The bacteria that exhibit gliding motility live in a variety of different environments, including marine, soil, and the human mouth. It really makes you want to go to the dentist and get those teeth cleaned, doesn't it?
Over the years, many different models have been proposed to explain bacterial gliding motility. Recent results suggest that it is unlikely that any single mechanism will be able to explain all forms of bacterial gliding. Instead, it appears that there are several different types of gliding motility motors. There are 2 types of bacterial gliding motilities: social and adventurous.
Social gliding relies on type 4 pili, such as the pili used in twitching motility. Pilus retraction appears to be the driving force behind bacterial twitching motility. The leading suggests that a gliding cell repeatedly extends pili from its leading pole, or the pole that is in line with the direction of movement. As the pili attach to a surface, they retract or pull back into the cell, resulting in cell movement. Cells could reverse their direction of movement by extending and retracting pili from the opposite pole. Oftentimes, some pili extend from the leading pole at the same time that others, which have contacted a surface, retract. This results in a smooth movement forward. Other filamentous bacteria appear to lack pili, which are responsible for the social gliding discussed earlier, and therefore other means of movement must be employed. Recent experiments have found junctional pores near the septa that separate cells of a filament. The pores are important in polysaccharide secretion. The polysaccharides extend from the pole opposite of the direction of movement and work to push the cell forward, leading to a smooth movement.
Numerous models have been proposed to explain gliding motility in bacteria without pili or polysaccharides. One model involves proteins or glycoproteins in or on the outer membrane that adhere to the surface. Other proteins in the cytoplasmic membrane and periplasm harvest the proton motive force and move the outer membrane proteins along tracks that may be fixed to the peptidoglycan. Think of a moving walkway at the airport. The motors must somehow coordinate their movements to result in efficient gliding. Another possible mechanism involves multiple conveyor belts composed of polysaccharide or protein fibrils that are moved along the cell surface. Movement of the proteins could result from export and import at different locations. Gliding mycoplasma cells are often described as flask or club shaped as above. The tip of the cell has a terminal structure referred to as the head or attachment organelle. Cells move in the direction of this structure. Mycoplasma cells exhibit a cytoskeleton composed of over 25 different proteins, which is thought to be involved in cell division, maintenance of cell shape, and attachment of cells to host tissue. The cytoskeleton may also be involved in gliding motility. Non-motile mutants have recently been isolated, and many of these have unusual cell shapes which may be a result of defects in cytoskeleton components. It's not well known how the cytoskeleton acts in mycoplasma gliding, but based on previous studies, we know that the cytoskeleton plays an important role.